Bismillahirrahmanirrahim. In the name of Allah, the most merciful and the most beneficent. Countless salutation on last Prophet Muhammad sallallahu alaihi wa alihi wasallam. So we were discussing about computational drug designing and structural bioinformatics. Mahtab Ali ji beta.
अस्सलामुअलैकुम सर।
वालेकुम सलाम.
सर मैंने आपसे एक क्वेश्चन पूछना था कि हमें ये जो स्ट्रक्चरल बायोइन्फॉर्मेटिक्स है सर, इसकी हमें ज़रूरत क्यों पड़ती है सर? इसकी हमें ज़रूरत क्यों पड़ती है सर, ये बता दें। मैंने लेट लेक्चर ज्वाइन किया था
अच्छा जी, मैं आपको बता देता हूँ.
स्ट्रक्चरल बायोइन्फॉर्मेटिक्स। थैंक यू सर।
Okay. So we have a question that why we need structural bioinformatics. So first of all, as we discussed in the start of this previous lecture, and we also have many lectures before, that what is the difference between conventional drug designing and computational drug designing. And if we don't have the expertise, resources and time for X-ray crystallography and NMR, then we have to use this structural bioinformatics approach. If we have Shah Naziz or GR here, please mark the attendance for last lecture and this lecture. And if they are not here, anyone else like Tehmina Latif, please mark the attendance. Thank you so much. So, Mahtab Ali, structural bioinformatics we need when we have to predict the three-dimensional structure of a specific target protein if the structure of that specific target protein is not available with the help of X-ray crystallography and NMR. So we don't have much time or resources. In that case, we just jump on structural mathematics approaches to structure the three-dimensional structure of that target protein, so we can know the structure. And as you guys know, function of the protein depends on the structure of the protein, and if you don't have the structure of the specific protein, you cannot understand specifically the function of that specific protein. However, you can have a sketch of the function of that protein from literature or from other experimental approaches. uh, However, you cannot specifically understand the function of that protein if you don't have the structure of specific target protein. So you have to predict the three-dimensional structure that lies in structural bioinformatics. After that, we have also the understanding of interaction of that protein with other proteins or biomolecules or drugs or DNA or RNA or nucleic acid. So for understanding of those, we also have to utilize the approaches of structural bioinformatics. And if we have to scrutinize millions of compounds from the library, and if we will follow the conventional high throughput screening approach, then it will take much time, and it also utilize or burn, you can say, many resources, and you also need experts of the domain for those experiments. However, for computational high throughput visual screening, you need less resources and time, and you can have similar sketch for that specific library or million of compounds for that specific target protein you are going to analyze against that protein, against that library or the compounds you have. So we need last time I sent an article titled as bioinformatics need of every biologist. How many students give a read to that? Please raise your hand. Okay, no one. It's very shameful and actual. So that's why you don't know the answers. Thank you, Aisha Makbul. So please read the recommended books or chapters or articles or given data. So it will help you to learn. If you have a look to that 2 or 3 pages article, bioinformatics need of every biologist, so there will be so many concepts in those 2 or 3 pages. Every domain or every field try to mention in those 3 or 4 pages. So please have a read to those 4 pages or 3 pages, so you can understand and you can have a sketch that in which domain specifically the bioinformatician we need. However, in every field nowadays a bioinformatician needed, so you should know where you should polish your expertise. I hope Mahtab Ali got his answer. And we were discussing about structural bioinformatics. First, we just have a look to the definition of bioinformatics. Like bioinformatics is an interdisciplinary field in which we use statistical, mathematical, and computational approaches to solve the biological problems. And usually bioinformatics has 30 to 40% success rate in experimental biology if the computational experiments perform very well. After that, we have a look to the definition of structural bioinformatics, in which we discuss about the prediction of structures, primary, secondary, tertiary, and the function of protein depends on the structure of protein. And if we have homologous protein available in the family, so we will use the structure of predicted homologous protein for the prediction of specific target protein. So after that, we have here tit- the topic software and tools. Anyone wants to give a read? Please raise your hand. जी आयशा मकबूल बेटा.
Open source bioinformatics software. Many free and open source
Better
software and
softwares and
tools are
tools.
available.
I think we didn't read in last lecture software and tools from here, please.
Okay, sir, sorry. Software and tools. Software tools, database, and servers from bioinformatics range from simple command line tools to more complex graphical programs and standard standalone web services are also available by various bioinformatics companies and public institutions. Open source bioinformatics software. Many free and open source software and tools are available through public and private institutes. While they continue to grow since the 1980s, still there is a need to develop new algorithms for the analysis of emerging biological problems. The open source tools often act as incubators of ideas or community supported plugging in commercial
In
application.
commercial applications. Thank you, beta. Thank you so much, Aisha. जी तहमीना लतीफ.
जी सर. Web services in bioinformatics. The basic in bioinformatics services are classified by the EBI into 3 categories: SSS sequence search services, MSA multiple sequence alignment, and BSA biological sequence analysis. The availability of these service-oriented bioinformatics resources demonstrates the applicability of web-based bioinformatics solutions and range from a collection of standalone tools with a common data format under a single standalone or web-based interface to integrative, distributed, and extensible bioinformatics workflow management system. Homology modeling. Homology modeling is a method to construct an atomic resolution model of the target protein from its amino acid sequence by utilizing an experimental 3D structure of a related homologous protein, the template. Homology modeling helps to identify one or more non-protein structures having resemblance with the structure of the query sequence. The sequence alignment aligns the similar and identical residues of the query sequence with the template sequence. The protein structures are more conserved as compared to protein sequences Amongst homologous but sequences falling below our 20% sequence identify identity can have very different structures. The sequence, alignment, and template structure are used to produce a 3D structure of the target protein.
Thank you. So there are two types of softwares for standalone softwares and web services. Any one of you just use Rasmol or maybe Modeler or these types of, there are desktop based softwares that you just download and use, and other softwares are web services tools, online tools that you have to connect with your internet, and then you have to connect with that specific software or tool to use to analyze your specific sequences or the target. So EBI just classified the services of bioinformatics in three main concepts, that are biological sequence alignment, multiple sequence alignment, and sequence search services. So you guys perform many times about MSA, that is multiple sequence alignment, in which you have more than one sequence or more than two sequences of similar protein, or you can say uh, same family protein, or one specific protein with many other proteins of homologous proteins or paralogous proteins for multiple sequence alignment, or you perform that experiments for to analyze the conserved region or conserved residues, or the residues that has similar physiochemical properties, to have a sketch that what is conserved in this protein family or the conserved domain of the protein family. So after that we have biological sequence analysis, that many biological sequences are available nowadays. There is a it is the time of COVID-19, and many researchers just working on the similarity of different strains of SARS-CoV-2 and the genome of that specific virus from different regions of the world, and then they just give a comparison through computational biology and multiple sequence alignment of the genomes of that different strains of COVID-19 or SARS-CoV-2 to analyze that where we have the similarities and where we have the differences from different regions of this world. Suppose they just sequence the genome of the virus from USA, Turkey, or maybe Italy, or Japan, China, Pakistan, India, Bangladesh, and then compare the similarity through multiple sequence alignment of those specific genomes of the strains they just retrieve from different regions of the world. However, nowadays researchers also working on the similarity and differences between SARS-CoV-2, SARS-CoV, and MERS, that are the same family viruses of coronavirus. That what is the difference and what is the similarity among all these different viruses of the same family, and if there are some conserved regions, and we already have the information of that conserved region with the help of MERS and SARS-CoV, so they can just utilize that specific information to solve this problem. So this approach of multiple sequence alignment or the multiple genome sequence alignment is also performed with the help of computational approaches and tools. If you will just Google, you can have many papers on this that the comparison of MERS, SARS-CoV, and SARS-CoV-2, and also the genome of different strains of SARS-CoV-2 from the different region of the world. And they reported the conserved region and the differences, and they also claim, or you can say justify somehow, that why specific region of the world has more death rate with because of this mutations or the changing strain of SARS-CoV-2, or the epidemiology or the behavior of the people or the environment of that specific region, or the immune system of the region of that specific region of the people of that specific region. So they just try to justify these. If you are interested, you can have a good read on these specific topics. After that we have homology modeling, and it is also known as comparative modeling. Through homology modeling or comparative modeling, as you know, the word is itself describing what is comparative modeling. To compare the protein sequence from one sequence to the other sequence, and the target sequence should compare with the other template sequences available in pdb or other databases if you have expertise in that. So you should have to retrieve the structure and utilize that structure as a template structure to solve the target protein three-dimensional structure. As we discuss it many times, that the function of the protein depends on the structure of the protein, and we can retrieve the structure of the protein or predict the structure of the target protein by utilizing the homologous predicted structures already available with the help of X-ray crystallography or NMR. And suppose if we don't get any homologous protein, then we will jump on any other protein that has good value or good amount of similarity and query coverage to select as a target protein to predict the three-dimensional structure of the to have the template protein to predict the three-dimensional structure of the target protein. So we have different steps of homology modeling. Anyone wants to give a read, please raise your hand. Two or three specific students just give a read. Everyone just try to read. This reading activity is just only for your improvement, so please contribute in this. Ji Shanaziz, after a long time you are here, welcome back.
Assalam. Steps of homology modeling. The homology modeling procedure can be broken down into four sequential steps: template selection, target template alignment, model construction, and model assessment. Template selection. The critical step in the homology modeling is the identification of the suitable template structure, if they are available. Template identification relies on the serial pairwise sequence alignments provided by the database search techniques such as FASTA and BLAST. Target template alignment. Homology modeling is based on the alignment of the target protein against the selected template protein. Template protein is an experimentally determined 3D structure obtained by X-ray crystallography or NMR techniques. BLAST and
Cluster W.
Clustal W are the highly cited alignment tools for the alignment of protein sequences. Model construction. The aligned tar- the aligned target sequence against the template protein sequence and the information obtained from the alignment employed to generate the 3D structure of the target protein is represented as a set of Cartesian coordinates for each atom in the protein (PDB) format. The model assessment validation. The validation of the predicted structure is another important step of the homology modeling. The evaluation of the predicted structure validates the accuracy of the structures on the basis of the experimentally designed structures. Numerous evaluation tools including PDB sum generate, Rampage, ERRAT, and Verify the 3D structure are Verify 3D are reported to validate the structures. The final selection of the predicted structures among all is based on the physiochemical properties of the protein, the cell type of the protein expression, and from the available data.
Thank you
Accuracy.
Shan, thank you. So there are sequential steps for the prediction of homology modeling. First of all, you guys perform modeler and homology modeling. Shanaziz, please, did you guys
Yes, sir.
perform homology modeling?
Yes, sir, we have done it.
With which software?
With Modeler, with HHPRED,
Okay,
and
and in which semester you perform this? In which course?
uh, in this semester, in your semester.
in this semester in my course?
Yes, sir.
Okay, thank you. So then you have a good look. And why you don't tell me before that you already performed this experiment? So in this experiment, as I remember, if you remember, so first of all, you should have to just retrieve a target sequence from UniProt, and after that, there are some steps for the prediction of homology modeling. That is selection of a specific target template protein from PDB. The structure should be resolved through X-ray crystallography or NMR, and after that you just align the target sequence with the template sequence, and there will be conserved regions. And if the similarity will be good, and similarity and QD coverage will be good, then you will have a very good structure, and you will predict the structure by using different softwares. After that, you have to evaluate the structure. Structure evaluation is very important to solve, to analyze the analyze the accuracy of that specific predicted structure with the help of Rampage, Erat, Verify 3D, What If, What Check, mole prob mole probability, and many other softwares are available, and every software has its own pros and cons, so you should know about the software. And I remember, if you guys uh, perform the homology modeling, then you guys should then you guys already have performed the evaluation. So we will now just have a good look to this chapter, because in this chapter we just discuss about the, okay, we have importance of homology modeling, why we just uh, use the homology modeling. We already discussed that homology modeling, how to retrieve the sequence from so we have to repeat it, or we just have to give a look in 2 minutes and finish this chapter? Shan Aziz, please represent your class. We have to study this chapter in detail, or we just have to finish this chapter as you are guys already perform homology modeling? Jishan.
Yes, sir. We already have done it, so we can just overview this chapter.
Okay, so we will finish it in 3 minutes. Thank you. So first of all, you just have to retrieve the protein sequence from UniProt knowledge base, and in this book RASF2, there was a protein RASF2 cancer protein that was utilized in whole book for performing all the experiments, and RASF2 was used to retrieve the sequence from UniProt. After that, so there is some steps how to retrieve the sequence, and after that you can say the homology modeling was performed, and solve the structure of that specific protein. Later on in later chapter, in maybe chapter number 4, we will predict the structure by homology modeling, threading and ab initio, and then we will have a detailed look on RASF2 structure prediction. So in this book we have computational drug designing, and then we just specifically have a look to a okay, now it's looking better, to a specific domain, that is structural bioinformatics. In structural bioinformatics, we just have a look that in this chart 7 different portions were classified. In first one, there is sequence retrieval and protein sequence retrieval through UniProt, and then similarity search through BLAST and cluster, and cluster W is used for the alignment of target sequence and the template sequence. After that, in chapter number 2, we have sequence analysis, in which sequence length, physiochemical properties of the that sequence, and prot param, prot scale, and pep states softwares were utilized to analyze the physiochemical properties of the selected target protein. In chapter number 3, structure analysis, we just have a look we will have a look to the protein function and active site analysis with the help of profunc. Then in chapter number 4, structure prediction will be studied, that will be used by web based tools, that is firex2, itusser, raptorx, swiss model, intfold to m40, and through homology modeling, modeler will be used, and how to perform all these experiments, and how these softwares work, and uh, interpretation of the results of the given results of those softwares will be studied in this chapter later on. After that, in fifth chapter number 5, structure evaluation, the predicted structure will be evaluated or validated with the help of IRAT, Rampage, Nolia, Mol Property Verify 3D, and many other softwares are available. You can have a look and use those for to evaluate the three-dimensional structure you predicted for your the specific target protein. In chapter number 6, it is a detailed chapter for the visualization and analysis of the predicted structure of the target protein with the help of UCSF chimera, in which structural elements, surfaces, labeling, energy minimization of the target protein will be studied. And in chapter number 7, that is the last chapter, molecular docking analysis will be studied, and how molecular docking analysis performed with the help of Autodoc tools, or Gold. Three softwares will be discussed, and we will try to understand how these softwares work and how to interpret the generated results of those softwares. If anyone has any question, please raise your hand. Okay, thank you so much everyone for joining.